Hello there, welcome once more to this YouTube channel. I'm glad you're there. Don't forget to subscribe to this YouTube channel. The topic is pharmacogenomics. This is a very vital topic in medical genetics for biomedical postgraduate students, particularly the Ph.D. candidates. We're looking at the course outline. So what are the objectives? The candidates should know how normal human genetic variability affects drug therapy. Candidates should learn how to apply pharmacogenetic knowledge in patient cases, integrating clinical and genomic data to drive clinical decisions. Candidates should understand ethnic as well as racial differences in drug response. They should gain pharmacogenetic knowledge to enable them use genomic data alongside other clinical data in optimizing clinical care. Now, what are the specific topics these candidates need to learn? 1. Introduction to pharmacogenetics. Secondly, you have cellular metabolism, molecular biology, and pharmacogenomics. 3. Drug metabolism, that's pharmacokinetics as well as pharmacodynamics. 4. Diversity of drug metabolism genes. They need to really understand cytochrome P450s, then the CYP genes. Next, in form of case study, they need to appreciate the pharmacogenetics of coagulation therapies. Next topic is pharmacogenetics of heart disease. They need to learn differential drug responses to cardiovascular disease medications. Case study of pharmacogenetics of hypertension. Then pharmacogenetics of diabetes. The drug therapies for diabetes should be taught. Pharmacogenetics of cancer with emphasis on the chemotherapy. Experimental genetics. They should know how to manipulate the genome. Then gene therapy with emphasis on expression vectors and transgene delivery systems. Next topic is pharmacogenetics with emphasis on stem cells and cloning in therapeutics. Last but not the least is pharmacogenetics and genomics, the ethical aspects. Right here is an illustration of gene therapy. When you have a disease as a result of a mutation or a defective gene, you correct the disease by introducing a favorable gene into the cell to take the place of the defective gene. And by so doing, you now have a favorable gene product being produced and the disease condition is corrected. Next, right here you have an illustration of stem cells and cloning in therapeutics. Cloning is an area that is currently thriving and is very important as far as therapeutics is concerned in the cure of certain conditions like cancers and all that. You could get stem cells. They could be embryonic stem cells. From the diagram you see, there could be embryonic stem cells. There could be stem cells from adults, like from the bone marrow. These stem cells are cultured and they are programmed to differentiate in the particular lines. Like the stem cells could be programmed to differentiate, for example, to give you cardiomyocytes and these cardiocytes. And the cardiocytes could, when they differentiate, they divide eventually. You could such cells could be introduced, implanted into the heart of someone that has heart failure or any other heart disorder, and such person will be cured of the condition. So that's a very vital area now. Thank you so much for listening. Don't forget to subscribe to this channel. Check out other videos on this channel as well for details of all these topics. Goodbye for now.